Hello everyone, welcome to the Gateway to Translation webinar. My name is Stuart Ellison and I'll be chairing today's session. So I'm looking forward to a very interesting talk from our guest speaker, Dr. Zoe Hewitt, on translating cell and gene therapies to the clinic. So just a bit of brief background about me. For those of you who don't know who I am, I'm a research fellow at the University of Manchester. With my expertise is in developing hematopoietic stem cell gene therapies for rare childhood genetic disorders. For those of you not familiar with the technology, it's based around taking patients' own stem cells, genetically modifying them with a lentiviral vector to introduce correct copies of the functional gene that's defective in the patients, and then giving these modified cells back to the patient where they can repopulate the blood system, traffic around the body, including organs such as the brain, where they can secrete functional enzymes to cross-correct the affected cells of the body. So I've been involved in developing 4 of these therapies for metabolic diseases, mucopolysaccharidosis type 3A, or MPS 3A for short, MPS 2, MPS 3B, and also another metabolic disease called Walman disease. I've been involved in developing the proof of concept studies and doing all the preclinical workup to lead these programs through to clinical trial. Our MPS 3A program is the most advanced at the moment, and it's been about 5 years since we treated the first patient. That program was licensed to Orchard Therapeutics, and we have some very interesting data from those patients. It's looking very promising. For our MPS 2 program, we treated our first patient last week, so we're excited to see how that develops. And currently at the minute, we're trying to work on developing a spin-out company to progress the MPS 3B program. And we're currently actively looking for investment for that spin-out. And finally, if any of that interests you and you want to contact me directly, you can find me on LinkedIn. It's currently my last week in Manchester at the moment. I'm actively looking for a new role. But my email will still be available as well, stuartellison-2@manchester.ac.uk. So that's enough on me. Moving on, I'd like to introduce you to our guest speaker for today, Dr. Zoe Hewitt, who is the co-founder and CEO at Regenerative Cell Therapy Consulting Limited. So Zoe has a background in pluripotent stem cells, which started in 2001 when she obtained a PhD from the University of Edinburgh. In 2006, she then moved on to the University of Sheffield when she established and managed a clean room facility responsible for deriving the UK's first clinically available human embryonic stem cells. And then in 2015, Zoe moved away from active research, becoming a project manager for the UK regenerative medicine platform, which focused on advancing regenerative medicines to enable clinical delivery and commercial development. This has led Zoe to co-found Regenerative Cell Therapy Consulting Limited in May 2022, which aims to facilitate the translation of laboratory research through to commercial development and investment. So without further ado, over to Zoe, and looking forward to hearing you talk.
Thank you very much, Stuart. And I'd like to start by thanking everyone for turning up today, and also for Alessandro, Giuro, and Ellie and Sally, I think it was, who have invited me to give this talk. So full disclosure, so my background is very much, as Stuart said, in pluripotent stem cells and cell therapies. But a lot of what I'll talk about is applicable to gene therapies as well, and actually to some of the combination med device, medicinal product borderline therapies too. So as Stuart said, my PhD back in 2001 was around developing strategies to eliminate undifferentiated ES cells from therapeutic populations, a topic that is still being discussed today as we enter a world of hyperimmune pluripotent stem cells and how we're going to eliminate any rogue cells. So definitely ahead of my time. My first postdoc, as Stuart said, was down in Sheffield. And I say postdoc in the loosest sense of the term because I arrived and the professor said, "We built a clean room, make it work." And my first response was, "You built what, sorry?" But indeed, we did make it work. I was responsible for developing the quality management system, for training all of the team. We had a fabulous team of technical staff who were just brilliant. And we made 14 brand new clinical grade ES cells in that facility and re-derived 3 research grade cell lines, one of which has been approved and in clinical trial. That was the first clinical trial in the UK, was the London Cure for Blindness. It was led by Pete Coffey down in London. And they converted SHEF1 cells into retinal pigmentosa epithelial cells and then put them into patients' eyes. That was on the BBC when Douglas said he was great chuffed to be able to see again, which was nice. And then, like Stuart said, I moved away from active research and was there at the beginning of the UK regenerative medicine platform right through to the end. And it was through that I then did the market discovery to establish Regen CTC, which I moved to full time just last year, with the idea that with over 20 years and frighteningly nearly 25 years now of experience in this space, there's got to be some information in my head that's useful to other people, given the fact that we've still only got 2 pluripotent stem cell or ES derived stem cell therapies come out of the UK, although iPS cells are making better headway. So I'm going to talk to you a bit about translating cell and gene therapies in general. And for most of you, that I've kind of sectioned this into 3 areas really. Most of you will be familiar with the preclinical work. In fact, Stuart's probably been through a lot of this pain himself. Target discovery, research and development, proof of concept will be very familiar to a lot of people. And on this slide, really, I'm talking about, these are the knowns, which for our academics are generally unknowns. So they're known unknowns or unknown knowns, whichever way you want to say it. But there's a lot of things here. And I think for me, one of the things that I want to highlight is there are a lot of things you have to do in the preclinical and early clinical trial phase. And these are really important decisions that will ultimately impact the commercial success. And one of the things that I hear a lot from academic researchers is, oh, we'll let people deal with that further down the line. And the problem with that is what we're seeing in the advanced therapy space is some of these products are becoming uncommercializable because of decisions, uncommercializable, because of some of the decisions that are made back in the research and development stage. So the more that we can do back in the early stages of these translational therapies or translational projects, the better, really. So this is a map that I've taken from the ATMP roadmap, and it's just really to highlight there is a significant development pipeline for advanced therapy municipal products, of which cell and gene therapy. And it's widely accepted now that these are much more significant. Well, these are significantly more complex than your average biologic. They are very diverse in nature, and frighteningly, the average time to go from proof of concept through to clinical trial phase 1-2 is about 15 years. And there are many things you have to do along this route. I'm going to share these slides so that you can hopefully access these links later. But one of the things that I would strongly advise is early links with regulators, but in those early discussions with regulators, really understand what your product is and know what you'll want to ask those regulators when you interact with them. Everything they will tell you will be case by case, but it'll be specific to the questions that they ask. They won't sit there and tell you how to do it. They will answer the questions that you ask them. So make sure those questions are well thought through before you go. Okay, so this will be a statement you'll hear probably quite a lot. Start with the end in mind. And for a lot of us as academics, we sort of start thinking about, well, who are we going to treat? What are we going to treat? Is there a real need for the treatment? And we think about that in the concept of what the standard of care is now and how we can better that standard of care. But I would also urge you to think about what you think the standard of care will be when you're ready to access the market. Who are you competing with? What are you competing with? Who is working in the space that you're also working in and may potentially have stopped working in that space? Can you unpack any of the reasons that they may have had for stopping that you can then use to learn from in your developmental pipeline. So that competitor due diligence, which is part of a lot of DPFS applications, for example, is really important because it shows investors and regulators that you understand the area that you're working in. So don't leave this, or what are we trying to do, end for as an afterthought. Really think about it really early on. Then think about why what you're making is better than what is either in development or already the standard of care. And don't just consider the cost benefit or the fact that there isn't a treatment and I'm creating one, but also think about how it will be delivered at the bedside in the clinic. What training will you need to give to doctors? Are they going to be able to have a very complex formulation at the bedside that you've decided is the best, or actually would a simpler system be more beneficial? So have an idea around that quite early on as well, or at least thoughts to consider it. This next bullet point is quite a big one. You need to think about where you're going to market your product. If you're going to go for global marketing, all of the jurisdictions, then there will be very different regulatory requirements that you have to consider early on. And it may be more sensible to target one area first and then have a parallel or second generation product that could target another, depending on how complicated that is, or take a step back and go a little bit slower so that you can address all of the regulatory concerns in parallel. That's less favorable often with investors, and so you have to balance the pressure of investors and wanting access to the clinic with being able to access all of the jurisdictions at the same time. So the final bullet point on this slide is you don't have to have all of those questions before you make the answers to all those questions before you make a start, but you should be thinking about them and having them incorporated into your developmental strategy. They could be possible lines of parallel R&D work that you need to do, where you could bring that data into your developmental pipeline as you transition. Okay, so for me, these 5 areas are common areas that people think about, but don't necessarily think about all of the things within them. So you have to make your decisions based around starting materials, how you select your contract development manufacturing organization partners, your clean room partners, how you identify appropriate raw and ancillary materials, the things you have to do around equipment and QC instrumentation. You have to have all of those decisions based in a science-based evidence framework, which is risk-benefit, which considers risks benefits to the product and the patients. You also need to have, with all of those areas, good documentation practice. It's essential that you write things down, but you also need to consider, as well as writing it down, can you recover it? Is there evidence to prove what you've written down? Can you justify why you're making those claims? And can you look at data in a way that you can trend differences as they come about? The other thing to say is that data from an investor or a regulator's perspective is not just your scientific results, which is quite often a misgiven. It encompasses holistically all of your data, so all of the traceability around all of your reagents, your starting materials, the suitability of those, how you've come to those justifications for using those products, which ties into how you document your risk assessment and decision-making processes. And all of that data package is what regulators, investors are looking for as you move forward. And then this goes back to what I was saying around commercialization before, circling back to the fact that you have to continually consider the end. You know, your decisions that you make in the R&D phase will absolutely impact the success at the end. And there are things where there is evidence now that shows it's not that we're bad at getting things into clinical trial. Actually, over the last 10 years, I think the number of clinical trials in the cell and gene therapy space is exploding. But the number that are going on to be first-line therapies in the clinic is actually quite small. And there are a number of companies that are dropping products, not because they're not safe, not because they're not efficacious, but they don't make money or they can't be commercialized at scale. Some of those are because of cell line choices or restrictions on IP and freedom to operate. So you really need to consider what you're starting out with and whether it's still going to be suitable to commercialize at the end. If that makes sense. It's weird not being able to see everybody's responses to what I'm saying. So if we break that down a little bit and we think about starting materials as an area of things to consider, in my space, the source of the pluripotent stem cells, so whether you're going to go for an embryonic stem cell or an iPS stem cell, that is a fundamental question depending on where you want to ultimately market your cell therapy. If you want to have an autologous or an allogenic product, again, you need to consider the source of your starting materials. And you need to think about not just the consent and whether there's appropriate consent to commercialize, but also the eligibility criteria that we use to select the donor. Have you undertaken due diligence of the clinics where that material is collected? Have you done due diligence of the documentation that supports the consent and the processing of those patient samples? That documentation should give you full traceability right back to the patient, and it should also cross-reference all the materials that were used. And that documentation needs to be kept for a long time, at least 30 years 30 years past the end of the product shelf life. So for pluripotent stem cell products, basically that's forever. Reagents have the reagents used in the generation of the starting material being suitably quality assessed. What are the risks associated with those? There can be risks, but you have to have systems in place to mitigate those risks and control against them. And if you don't know what they are because you didn't ask the questions, you can't do that. Do you have the freedom to operate with that source material, that starting material? That could be IP restrictions, it could be consenting restrictions. So you need to have looked into that fully. That then leads to, do you have the appropriate licenses to use them? And approvals in place, and they differ whether it's gene therapies or cell therapies, as you know. And again, this final point comes again back to the whole decisions that you make in research impacts the commercialization. When you're thinking about the cell therapy or the cell starting material, you need to think past the first in human trials. So a lot of academics, when they're thinking about translating research, they think about first in human clinical trials, which are essentially safety trials. But the requirements as you go up through the trials at phase 2 and phase 3 become much more stringent. And so you could ultimately get to first in human trials. And at best, you will have to go back and do some comparability testing if you change your starting material, for example. At worst, you could be looking at a whole new product submission for the regulators. So it is a big risk if you've not got that correct. So again, for cell therapies, but also for your starting material for making gene therapies, there will be a level of consistency of it would be an ancillary material, I guess, for gene therapy because it's the expression vector, the expression system. So for cell therapies, are you doing allogenic or autologous cell therapies? And the requirements for cell banking differ. Are you engineering your cells, and are you engineering them before you make your master cell bank or after you make your master cell bank? Are you engineering on a patient per patient system like in iPS cells? That will impact when you might need a clean room. So there are different feelings in the regulators. I don't think they've nailed this down yet, but there is some thoughts that you would have to do any genetic engineering within a clean room. That then puts the cost up. If you're making a single genetic edit and then you're making your master cell bank and that edit would be in every subsequent cell therapy, then there is the possibility you could risk assess doing it outside of a clean room. But you would have to have all of the quality management documentation in order to mitigate those. And again, with the documented justification for your decisions. Consider also the tech transfer process. So from your research lab, you will have to partner with a contract manufacturing organization with a clean room. There will be a time element in transitioning from your research lab to the CDMO partner. Having those procedures, testing them on your peers in your lab to make sure that they are unambiguous is very important and helpful. Again, the reagent quality and looking at what QC testing will be necessary to prove that reagents you're using that have been determined as critical reagents for your process have been thoroughly thought through, and you can prove that when you're banking as well as when you're making a final product, are essential. Think about the size of your bank at the start. So if you're going into a clinical trial, what you don't want to do is have a master cell bank that's too small, so that when you come to your phase 3 clinical trial, you haven't got enough material to make the product that you want. So you also have to make sure that there are enough samples in there to do all of the relevant QC testing, the retention stability study samples, all those kinds of things. That will also impact your fill finish format. So there are some limitations at the moment with vials versus bags and how much you can fill. So that depends, and the equipment that you need to fill it in an aseptic way. So that also impacts the decision around making your cell bank size. And again, documentation, traceability is fundamental. I think I've talked quite a lot already about reagents, and I guess I've put this in because if the reagents that you're using are not likely to be in your final product, they would be referred to as ancillary materials or raw materials rather than starting materials. So the starting material for a cell therapy, if it's a pluripotent cell therapy, then that material will still be part of the end product. Whereas things like culture media, matrix coatings, cryopreservation agents, for example, they're an integral part of the manufacturing process, but they don't form part of the product. But you do need to control them because they have a direct impact on your product or your therapy. Similarly, if it was a gene therapy. Consumables, again, they are not part of the final product, but they do often have a direct influence on your product, essentially, especially things like cell culture plastics, but also cleaning reagents. They're not directly connected, but they could potentially impact the environment in which the product is being made. So it's important that all of the raw materials, ancillaries, consumables are risk assessed and assessed not just for contamination risk to the product, but also for their impact on a manufacturing process run failure, for example, because you don't want to be throwing away batches of material. So how reliable are your manufacturers of consumables to make a consistent cell culture surface, for example? That doesn't mean half of your cells don't stick down, or you've got leachables from your bioreactor bags that mean that half of your cells die in the manufacturing process. So it's those kinds of considerations. And just because a company will say it has GMP reagents, don't take that as being read that they will be suitable for clinical use. There's a lot of confusion, I think, around what GMP means. You could make a component for a car in a good manufacturing practice way. It doesn't mean that you would embed it into somebody's body in the same way. But you should be auditing suppliers either physically or at least through questionnaires so that you can identify risks with your suppliers. And again, they don't have to be risk-free, but you have to know what the risks are so you can put in controls and strategies to mitigate against them. And it's those controls and mitigations that the regulators look for to show that you have a good understanding of your processes. Moving slightly to the side of this area, so the equipment that you use directly in manufacture, but also the instruments that you use for quality control, they've got to be shown to be fit for purpose. And in a regulated environment like a GMP contracts manufacturing organization, they would be designed, installed, operate, and perform in a certain way, and that would be qualified at the beginning of the process. But in a research lab, as you're starting to think about this, you should think about the equipment and the instruments that you're using, and have a good understanding about the instrument drift. You know, how long in between calibration can you guarantee that you'll get the same measurement every time? So understand what a failure looks like and how failure recovery is undertaken, and what the impact of a failure of part of a process would be on your process. You should also have maintenance and cleaning records that you can keep. It's a good habit to get into because you will need it later. So now I'm taking a bit of a step forward into, you're now starting to manufacture your product. Now for cells, there could be an intermediate step in your process. So you would end up with a drug substance rather than a drug product. So that intermediate step could be fresh or frozen still, but it could be a progenitor through a differentiation process that you then thaw again. You freeze it down, you thaw it again, and then you mature it, and then that's your drug product. It could be a suspension that you have to seed, for example, onto a scaffold or a delivery device. So that's the difference between a drug substance and a drug product. You need to consider at the drug substance stage still, what are you going to store it in, whether those are suitable for clinical use, what media you're going to put it in. Do you have any idea of how long it is stable for, if that's fresh or frozen, and how are you going to ensure that it's labeled correctly? So things that you also have to consider. When it becomes ready to be released by a qualified person, you know, when the QC has been done, then it becomes the drug product. If it then needs combining or seeding onto a scaffold or a matrix at the point of transfer into a patient, and that would be classified possibly as formulation, depending on how complex that is and the length of time in between doing that and administering it to the patient. At this stage, you also need to be considering how you're going to do these different releases. So how are you going to do a QC release for an intermediate drug substance? How are you going to define that a product is ready for QP release? What are the analytics? What are the standards that you're going to use? Are those assays validated? Do they show potency? Is it quality? It should be all of those things, but these are the sorts of things you need to be considering. And then finally, the formulation. I've already talked about the formulation. I don't know what I was talking about before, sorry. The formulation and the storage, transport, and whether your delivery into the patient can use an already CE marked device, and whether that device needs to be certified for a different product, or whether you need to develop a new device. There could be a level of interface between your delivery system and your drug product that you will have to do before you get to clinical trial, that you need to consider early. Regulation, I'm not going to talk about massively because it's massively complicated depending on your specific product and also the jurisdictions in which you're going. But to say that you need to consider the regulation in the jurisdiction that you're manufacturing and also where you want to sell. It can impact the classification of what it is you think you're making, depending on the regulator. As I've already said, be specific with your questions when you do interact with the regulators. Do it early, and be aware that it's case by case, the advice that they often give. The regulators are always looking to make sure that you've done risk-benefit analysis, and they're looking for evidence of your decision processes and your risk mitigations. If you haven't written it down and you can't evidence a decision, they're likely to dismiss it and ask you to go and do that. So this is a common statement that I hear. We don't need to do quality management. You only need to do that if you're making a product and you're going into regulatory submission. And yes, evidence of quality management is expected when you do regulatory submission, but no, that's not the time to start considering it. If that's the time you're starting to consider it, you've missed the boat, and you'll have a lot of backfilling to do when you come to do your submissions. Early implementation of quality management enables you to have evidence, and it supports traceability of all of the steps of your manufacturing process. It gives you better investigative power when you're at the R&D stage. It saves you time, it saves you resources, it saves repeating unnecessary experiments, and it just shows regulators and investors and TTO offices and people who you want, grant bodies, even publication houses, when you're trying to get your work published. It shows everybody that you've got good science at the end of the day. And at its heart, it reflects 4 simple principles: control, traceability, transparency, and responsibility. And its purpose is to drive continuous improvement. Quality management principles are not just regulatory needs. They are generally just good operational and business principles to adhere to. And actually, if you implement some of the basic ones early, they become a bit addictive. Maybe it's just my personality, but they do become a little bit addictive. And quality management isn't a new phenomenon. It's not something that we invented a couple of years ago, or even it's been around since medieval craftsmen in the 13th century. They used to have guilds, and it was the responsibility of the guilds to set up rules for products and service quality, and then they had inspectors, and then they were marked if they were flawless. And in the end, the craftsmen added their own mark, and then that became as a mark of a good reputation. So it stood the test of time until really the Industrial Revolution, when things went a bit Pete Tong, because I shouldn't say that. Mass production became the goal, the primary goal, and the quality took a dip for it, really. But it wasn't. It was actually the mid 20th century, when, from the Japanese engineering industry, that the modern day sort of company-wide holistic quality control really arose. And actually, it was 1987, so younger than me, when ISO 9001 quality management system was first developed. So quality management has been around for a long time, and we should be good at this by now. And it's important because it stops things like the thalidomide incident in the 1960s. This is really when the Medicines Act came into force, because this sedative was repurposed basically, and said, yeah, it's safe for everybody, but there was no data to back that up. And it's not the only one, and it won't be the only one. The purpose of quality management, as I've said, is you capture when things go wrong, but you know why they've gone wrong, so that you can put things in place to improve it for the future. And there's, it's constantly evolving as we make medicine safer and safer. So potential solutions was also part of the remit for today's chat, really. So where do you start? It sounds quite daunting. And I think I would start by saying that from an R&D perspective, no one would expect you to have a full-blown quality management system in a research lab. You don't need one, and it's something that will evolve with you as you develop products to head towards a translational goal. But I would recommend, or Regen CTC would recommend, you start by implementing 4 fundamental founding principles or elements: good documentation practice, quality risk management, event reporting, and change management. That was the only image that, sorry. It was the best image that I could find for change management. I don't expect you to all be changing nappies. So for me, lab books are not effective quality management tools. They're something we all use. Some people are better at completing them than others. If they're used well, they can support due diligence, but only if we standardize training to use them. The number of times I've seen PIs particularly hand a blank A4 lined notebook to a student and gone, here's your lab book. and then they're left to it. No one tells you what to write in it. No one tells you why you've got to write the things in it. And as somebody who spent a good portion of her career going through lab books to try and find vital bits of information, they are essentially not worth the paper they're written on vast majority of their time. And there's some, the training doesn't have to be onerous on this. It doesn't have to be hours of lectures and modules on how to complete a lab book or why it's important to complete a lab book. But it should be consistent with every new member of staff, and it should be reviewed to see if it's effective. And I would say that actually you could eliminate a lot of the problems by just giving new starters a checklist of the types of evidence that must be recorded in a lab book. Better still, create some templates for standard assays where they have boxes to fill in for things like lot numbers of reagents, or a place where you have to write if something went wrong, or the version of the SOP that you were using to run the assay, so that later you can go back and check if something is different, why it was different, and you can tie a cause and effect. Alcoa is something that absolutely should be implemented early. It's a fundamental GMP principle, but actually it can be used at any point. And I don't normally at this point, I would ask if anyone knew what Alcoa was, but given that everybody's microphone's off, I won't. But this is a way of making sure that data that you record in said lab book is attributable. So it's identifiable to a person, and you can identify that person, that it's legible, and that's not just, black and blue pen now, but that it's always legible, so that if you photocopy it, it's been photocopied and it's not original, that you don't use pencil, you don't rub things out, that it's contemporaneous. I hate that word. I always struggle to say it, but actually, and again, this would normally be a participation question, how many people, show of hands, or think about it in your head, have written the fundamental concentrations of things on their lab glove and then copied it up into their lab book later? Me did that before. Everybody, I think, probably, if you're honest. But it's not contemporaneous, because you could technically have made a transcription error at that point in time. And so actually what you've done is falsified data. What you should do is stick the glove in your book and then write out next to it, so that you can compare the fact that you didn't make a translation error. Health and safety would not like that, because obviously gloves are not to be kept and shouldn't be in lab books. But there is a problem here if you don't write things down at the time and you copy them up later. And again, it wouldn't be original if you've copied it up. So, you should be retaining original results in your lab books. It doesn't matter if they're untidy, but it does need to be legible. They should be accurate. And any errors that you make, you should correct, but you should be able to see the original entry. So a single line through it with your initial, and then you justify why you're making that change. The golden rule with all of this is, if it isn't written down, it didn't happen. The other important thing about documents, and the other bugbear with lab books, is lab books are it's a diary, essentially. It's chronological. Retrieving information from lab books is actually quite difficult, and especially in cell and gene therapies, when they're developed over a 15-year period. You think about academic contracts where everyone's got maybe 2 years here, 6 months there, maybe a 3 or 5 year if you're lucky, but that's a lot of people and a lot of different ways of storing and recording data, and the PI at some point is going to be responsible for retrieving it all. So the onus really is on putting in systems where you can retrieve data and you can trend data. So as I've said, these are long, complex project development pipelines for these products with lots of data. We've got short contracts, and knowledge retention when people are leaving is challenging. But if you have training on this and you've got all of this information in one place, it will improve training new staff, and it will also improve the rate at which we progress, because all of the information will be on one place. And if it's easy to look at, people will look at it before they go, well, I know what I'm doing. And then you will not get so many repeats of experiments that don't work. So again, my solution here is develop operating procedures, write them. The people who are performing them should write them. They should test them on colleagues, get colleagues to look at them to see whether they are clear and unambiguous. You often write in a way that you know what you're talking about, whereas the person next to you probably has got no idea. So write them and have them tested. I would also suggest using project files or folders rather than lab books. E-lab books maybe are helping now, because you can put file structures into e-lab books where you can have multiple contributors to a project. Use a predefined filing structure, again, that's trained into every member of your lab. Use defined file names and formats for file names with good metadata, and then that makes looking for the records that you need with the information in easier, because you can search through your OneDrive or Google Drive or whatever it is you use. Always cross-reference file locations in lab books. So another bugbear is you go to a lab book and it says it's filed electronically, but it doesn't tell you where. It's not helpful. Okay, so that's the first founding principle. I believe everyone in the room should be able to implement good documentation practice, and it would massively improve even your thesis write-up, if not a publication, but it would massively help. Quality risk management is the next one, and this for cell and gene therapies need to consider the risks to the clinical use of those cell and gene therapies. So it's quality in terms of the safety and the efficacy or the potency of the cell and gene therapy. Your decisions around quality risk need to be data-driven, again, evidence-based, risk-benefit assessed. And you need to be thinking about how you can reduce any risks back to acceptable limits. Quality risk management is fundamental because there are a number of other processes within quality management as you progress through to a GMP facility, for example, like deviation reporting and change management, that need to consider the risk of making a change or the risk of an event. So having that risk framework established early on is very helpful. It doesn't have to be complicated. We've all seen these risk matrix before. We're usually using them from a health and safety perspective, whether I've got a minor injury or I'm going to be off work for a number of months. But it's the impact of what you're doing on the product that you're making at the end. This is an example of one where we weighted the severity of the impact over the probability. And you should have them defined. So defined both in what impact means for your product, and also put timeframes in the probability. So in this example, it was a sort of 3 month. If something had occurred within 3 months, that became a problem. If it was further away from 3 months, then it was less of a problem. That may vary depending on the speed at which your manufacturing process occurs. Again, I'm not going to dwell on these because I'm going to provide the slides. So I very briefly mentioned then event reporting. Defining protocols is one thing, but ensuring that they're robust and they're being used correctly is something completely different. It's an essential part of quality management is to have effective event reporting. It supports the ability to investigate and improve processes both efficiently for the staff, but also the safety of the product. It requires a strong leadership and an active learning positive culture, not a blame culture, and that will then engage staff to report when they've done something differently, and it empowers them to make improvements. So we defined an event as an action or an occurrence which differs from what is expected or written, irrespective of impact. And we did that on purpose so that everybody would report everything. So you get all kinds of things reported when you have such a wide definition. You know, I dropped my flasks on the floor and they've all got bubbles in them. That could be important if they also developed a crack and that was the cause of the infection that you then got. We had things like I got the numbers round the wrong way and actually made this concentration, but I noticed it. No impact, but at least you can record it. most events are unintentional. Most people are not doing things on purpose, but nevertheless, small changes to a process that you didn't intend could have a big impact. And if you get into the habit of recording these, you can just streamline processes. It helps. Near miss opportunities, I think, are the most important thing, because they're learning opportunities, something that didn't happen. It also allows you to validate the control mechanisms that you've got in place. So if you had a near miss, you probably don't need to do anything about it, as long as it was detected by one of your actual monitoring steps that you intended. The other important thing with event reporting is to make it easy. So have logs so that people can write it down in real time in the lab, but make sure you look at them, because no one hates it. No one hates more writing things down when they're not then looked at, because it seems like a pointless exercise. Conscious of the time. So change management, I'll quickly go through this. It's a key element. It should be stage appropriate. At the beginning of the when you're doing R&D and you're thinking about early translation, you don't want your change management process to be too onerous, because you do need more flexibility. You are doing research at the end of the day. But scientific research is built on controlling variables. If you don't control them, or if you don't record them, if you've made a change, then you're not doing very good science either. So I'm rushing through this because I'm conscious of the time and I want to leave time for us to have a conversation and a discussion. But with change control, what you perceive to be an insignificant change could have a significant difference later down the road. And also, changes that you think are obvious will probably be obvious to the next member of staff who comes onto your project if you leave. And therefore, if they're not all written down in one place, what happens for the PI is the same experiments are being done over and over again, and you're wasting resources. Whereas if it's easy to look and say, okay, this has been tested and this has been tested and these are the impacts. So a simple spreadsheet is all you probably need, I think, but at an early stage. As you progress through the developmental pipeline, you will have to lock down a process, and therefore you really do need to consider the implications of a change before you make them. And I think before you make them is the important part with change control. There's no point considering the impact of something you did differently afterwards. That makes it an event because you haven't followed the procedure. Whereas if you can think about how is making this change to the process going to improve this, or if I don't make this change, what's going to be the negative impact of not making this change before you make it, then you can evaluate the success of the change rather than evaluate the impact of something. I need to flip this on. So when you only have one person working on a project in R&D, it's perceived that the change has no implication. But actually, if that person leaves and they haven't recorded the changes they've made to an SOP, then somebody else comes in. It's just a lot of repetitive work. When you start to move towards multiple people on a project, then you have a communication issue as well as just a change control issue, because you need to be able to tell everybody that this process has now changed, and you train everyone into doing it. It will be a formal part of clinical trials is having a locked down protocol that you shouldn't be changing because all of your safety data is related to that. So therefore, I would suggest that's why I'm suggesting it needs to be stage appropriate. So just to finish off then, sorry, I've rushed through that a bit at the end, but I want to have time for a discussion. The cell and gene therapies are complex. They've got long developmental pipelines. There are many challenges, but many of them are known, and they are similar across the different modalities, and we can learn from each other. quality management principles aren't new, and they can be applied very early in innovation and through translation, and they will benefit you enormously. They can empower your workforce to help make changes for the better, as long as they're recorded. But you do need to have engaged leadership, because you have to show from the top that this is the way that you want to operate, because if you're trying to do it from the bottom, you never get there. A QMS system is never finished, and it should grow and improve with your product and your team, is the final thing that I would say on that. So apologies that was a bit rushed at the end, but I do want to have a chat, and I've seen a few things come in online. If you want me to talk any more about my desire to have everyone operating in a quality management environment, then I'd be happy to. So thank you.
Well, thank you, Zoe, for a very interesting talk there. We've got